Hello, everyone. Thank you for joining us today for our webinar, Osmotic Shock and Cell Count Accuracy, Ensuring Accurate Cell Density and Cell Viability Measurements. But first, some quick housekeeping. Please feel free to submit questions that will be answered during our Q&A session at the end of this webinar using the Q&A function. Please note that today's event is being recorded and will be sent to you within 24 hours of the conclusion of the event. Now I would like to introduce our speaker for today's event, Shweta Nair, Director of Bioprocessing at Nova Biomedical. Shweta is the Director of Bioprocessing at Nova Biomedical with over a decade of experience in the biopharmaceutical and life science industries. Since joining Nova in 2019, she has led the development and launch of innovative analytical technologies that improve accuracy, reliability, and data integrity across the bioprocessing workflow. Shweta drives product and business strategy through strong customer partnerships and a deep understanding of evolving industry needs. A collaborative leader, she is committed to building high-performing, cross-functional teams and translating customer insights into impactful solutions. Prior to Nova Biomedical, she worked in global roles at MilliporeSigma, managing laboratory equipment for biopharma and research applications. With that, I will turn it over to Shweta.
Thank you, Jared. A very good morning, good afternoon to all of you. Thank you for joining us today on this webinar on osmotic shock and cell count accuracy, really ensuring accurate cell density and cell viability measurements. So let's get started. As Jared mentioned, I also want to reiterate, please feel free to type in your questions that you may have for us using the Q&A function that you'll see on the top of your Teams tab. We'd love to hear from you. And if we cannot get to the question during the session, we'll make sure to follow up with you based on what you put in there. So please feel free to type those in as we go through. I would also like to take a moment to give you an overview of Nova Biomedical. We're a global life sciences company focused on increasing productivity and ensuring quality across regulated laboratory environments, including bioprocessing, clinical diagnostics, and food and beverage. We support customers in more than 90 countries with a very strong focus on technologies that really streamline workflows while delivering confidence in results. With that context, I'd like to talk about a recent news with regards to Nova Biomedical, as well as an impact that has for the larger life sciences company. Advanced Instruments and Nova Biomedical came together in 2025, July of 2025, to form a unified life science organization focused on these regulated laboratory workflows. With the two companies coming together, they really bring decades of expertise in biosensors, osmolality, automation, and bioprocessing, forming the foundation behind the Bioprofile portfolio, which includes the Flex 2 and the Fast CDV, and its reliable day-to-day use in cell culture monitoring that we will be speaking of today. Within Nova Biomedical, across bioprocessing, we support customers throughout the workflow, right from cell line development, upstream process monitoring, downstream support, and quality control. Within bioprocessing, we offer several complementary portfolios, including the Solentin portfolio for single-cell cloning, Osmotec portfolio for osmolality testing, and Artel for liquid handling verification. Today, however, we're focused on our Bioprofile portfolio, which is centered on routine cell culture monitoring. Bioprofile Flex 2, our flagship product, provides a comprehensive multi-parameter analysis for cell culture health, while Fast CDV delivers rapid, automated cell density and viability measurements. Together, they really help teams get the right data at the right time without adding complexity to the lab. Let me start by giving you a quick overview of what we'll cover today. First, I'd like to walk through the evolution of Nova Bioprofile products, with a focus on how our cell counting technologies have developed over time. Then we'll shift into the main topic, how we've uncovered a key issue related to osmotic shock caused by manual sample pre-dilution and the impact that has on cell count accuracy, cell viability, and cell density. From there, I'll share insights from our almost year-long investigation, including the steps we took to test and validate what we're seeing. After that, we'll talk about some important design considerations to keep in mind when selecting an automated cell counter so you can achieve accurate and reproducible results in your lab. So with that overview of today's agenda in mind, let's start by taking a step back and looking at how this technology has evolved over time. Nova Biomedical introduced its first bioprofile analyzer back in 1998. And at that point, it was a relatively simple system. It was focused on measuring pH, gases, basic chemistries. As customer needs grew and processes became more complex, there was a clear demand for more integrated solutions. In response, we introduced our first automated cell counter as part of the Bioprofile Flex 1 in 2008. The goal was to bring everything together. One instrument, one sample, and all the critical cell culture parameters that you would need. So labs no longer had to rely on multiple separate systems. We continued to build on that foundation with the Bioprofile Flex 2 when we launched that in 2016. And most recently, the Bioprofile Fast CDV cell culture, cell viability and cell density analyzer. Across all of these platforms, we've used image-based analysis to measure cell density along with trypan blue dye exclusion to determine viability. But what really differentiates the system is a key design feature: onboard automated dilution. This allows samples to be analyzed directly without any manual pre-dilution at concentrations up to 160 million cells per milliliter, covering virtually all applications, including your high-density perfusion cultures. And this feature isn't just a convenience, it's actually what led us to uncover a critical issue that we'll delve into next. Before we speak about osmotic shock and how it can be significant to your count and accuracy, I'd like to explain how we uncovered the impact of osmotic shock. First, I would like to talk to you about how the bioprofile systems are typically implemented in a lab setting. When a customer purchases an analyzer from us, an installation is completed by our field service engineers and our applications team. They work very closely with the customers to run what's called a comparability study. The objective here is very straightforward. We optimize the analyzer's algorithms so that its results align with the customer's existing reference method. So we want to make sure it aligns with what data you've been getting so far from your reference analyzer. We can do this across all measured parameters, pH, gases, chemistries, but the most common focus is on cell density and viability. With many different automated cell counters in the market and our very own journey in this space, particularly with the Flex 1 and Flex 2, customers have consistently requested alignment to their established methods. As a result, we've built over a decade of very intimate experience aligning our technology to a wide range of reference analyzers. So how does this process actually work? It starts with a scheduled on-site visit from our global application scientists. So no matter where you are, there will be an application scientist from our team that will be able to help you. And it's, again, no charge. Ahead of the visit, we coordinate with you to ensure that that all the representative cultures are available. That means covering different cell lines as well as a full range of conditions, low to high cell density and high to low viability. During the study, we typically measure 7 to 10 replicates across these conditions: low, medium, high cell densities, early stage cultures and high viability, and late stage cultures with lower viability. The goal is to fine-tune the analyzer so that the single cell inspection type can be used across all stages of the process, from scale-up through production, while maintaining results within about 10% of the reference method. Now, the phenomenon I'm going to talk about today showed up during several of these comparability studies that we performed at customer sites. In this case, our applications team was working with a customer and collected CHO cell culture samples panning both early stage and late stage production. For each condition, they ran 10 replicate measurements on both the Bioprofile Flex 2 and the customer's reference analyzers, which was a very commonly used automated cell counter analyzer based on trypan blue dye exclusion. Also, just to note, because of confidentiality, the data I'm showing here just was generated with our university partners using a non-transfected show parent line. That said, the trends you'll see here are very consistent with what we observed in the original study. So initially, everything looked very encouraging. All low cell densities, the agreement between the two systems was strong. But as cell density increased, we started to see something unexpected. Once we moved above about 10 million cells per milliliter, the results began to diverge. What had been within roughly 10% at lower densities became increasingly different as the concentration went up. And right around that 10 million cells per milliliter mark, there was a very noticeable break in the trend. It may be a little difficult to see in the full data set on this slide, so I'm going to zoom in a bit more to focus specifically on the high density samples, those at around that 19 million cells per milliliter. Here, when we zoom in on those high density samples, the pattern becomes much clearer and honestly a bit more concerning. You can see a distinct downward trend in the reference analyzer data, which is shown in red. In contrast, your Bioprofile Flex 2, shown in blue, remains very consistent, with strong reproducibility across all 10 replicates and no meaningful drop in viable cell density. At this point, this simply didn't make sense to us, so we launched what turned into a long investigation to understand what was driving this behavior. To do that, we took a close look at how the samples were being handled, because that's often where subtle issues can arise. In this case, we prepared 10 replicates for the Flex 2 and 10 for the reference analyzer. Since these were high-density samples above 10 million cells per milliliter, we had to perform a 1:4 pre-dilution using PBS to bring them within the measurable range of the reference instrument. All of the samples were prepared and diluted at the same time, then loaded onto the tray. From the first measurement to the last, there was about a 30-minute window. Now, it's worth emphasizing that on the Flex 2 data itself was acceptable to the customer, right? The challenge was the inconsistency we were seeing in the comparison. What we started to notice was that the results seemed highly dependent on how long the samples sat after dilution. If the samples were analyzed quickly, the agreement looked good. But as that time increased, the results began to drift, sometimes by as much as 10 to 20%. And that's where things became really challenging. We weren't dealing with a consistent bias that could simply be calibrated out. Instead, the variability itself was changing depending on timing. That made it nearly impossible to reliably optimize the algorithms or achieve stable alignment between the Flex 2 and the reference method. And that inconsistency is what ultimately led us to dig deeper into the underlying cause. So as with any investigation, our very first step was to step back and map out all the possible variables that could be driving this deviation. We started with the usual suspects: pipetting variability, inconsistent mixing or vortexing prior to dilution, and general sample handling. And just as a reminder, these manual dilution steps are only required for non-NOVA biomedical cell counters. With the NOVA biomedical systems, like the Flex 2 or the Fast CDV, all dilutions are performed on board and automatically. We also looked at the materials involved. So for instance, the Flex 2 and the reference analyzers use different sample cups. So we asked whether cell adhesion to the walls could be a contributing factor. Since the trend we observed was a decrease in measured cell density over time, that seemed plausible. The longer the sample sits, the more cells might adhere to the surface. Another area we evaluated was instrument mixing. Different analyzers use different mixing routines, so we considered whether incomplete resuspension, especially of the cell pellets, could be leading to a lower reported cell densities. At the time, this actually seemed like the most likely explanation. But after several months of testing, none of these factors fully explained what we were seeing. What did stand out, though, was a consistent pattern. This phenomenon only appeared in samples that had been manually diluted. To probe that further, we began diluting samples even below 10 million cells per milliliter, and we still observed the same downward trend in about half of those cultures. So that raised an important question. What was different about those samples? At that point, we shifted our focus from a process variable to biology itself, what might be happening at the cellular level, and one key difference emerged was osmolality. Higher density cultures typically have higher osmolality due to accumulated feeds and pH control additions like sodium carbonate or bicarbonate. In contrast, most labs diluted samples using PBS, which has an osmolality of around 300 milliosmol per kilogram. So we started to ask, could this sudden change in osmolality, this rapid shift in cells environment, be responsible for the effects we were seeing? At this point, we had begun forming a hypothesis. We considered whether the sudden change in osmolality and the basic physiology of passive water transport across the cell membrane could be driving what we were seeing. Specifically, could cells be rapidly taking up water to equilibrate intercellular and extracellular osmolality? If that were happening, it could lead to a measurable increase in cell size. So the first question we asked was, can we actually observe a change in cell diameter as a function of the difference between the culture osmolality and the diluent osmolality? If we could confirm that, it would support the idea that rapid cell swelling might eventually lead to cell lysis and ultimately a loss of viable cells in the sample. To test this, we designed a controlled experiment. We started with cell culture samples at approximately 300 milliosmol per kilogram. Then we prepared 5 different diluents by combining trypan blue with a stock sodium chloride solution, creating a range of diluent osmolalities from 250 up to 450 milliosmol per kilogram. Using those, we generated 5 separate samples at a 1 to 2 dilution, resulting in final osmolalities between roughly 280 and 300 milliosmol per kilogram. We then analyzed all of these samples on the Bioprofile Flex 2, and the measured cell diameters are shown in the graph here, as you can see. The changes in the diameter, if you notice, are quite significant, even with relatively small shifts in osmolality. Now, it's worth noting that this setup is somewhat the inverse of what typically happens when you're doing your routine sampling. We designed it in this way intentionally to really eliminate variability that could come from pulling samples from different bioreactors with different baseline osmolalities. But the takeaway here is still valid and clear. A rapid change in osmolality leads to an almost immediate change in cell size. So now if we were to go back to our original data set comparing the Flex 2 with the reference analyzer, the question is, why didn't we see the same downward trend in viable cell density with the Flex 2? And the answer comes down to timing, right? While the Flex 2 does perform dilutions using trypan blue and when needed an onboard diluent, the key difference is how quickly that process happens. The dilution occurs immediately before analysis. The measurement is completed within seconds. Because of that, the cells don't have enough time to fully respond to the change in osmolality. They haven't equilibrated, and more importantly, they haven't reached the point where swelling leads to cell lysis. This behavior is consistent across all of our cell density modules and analyzers, and it's a major reason why the results remain stable and reproducible. Now, if you contrast that with traditional workflows that rely on manual dilution, the impact can be significant. When samples are diluted now manually, there is often a delay between dilution and analysis, sometimes several minutes or even longer, depending on workflows. During that time, cells are exposed to a new osmolality environment, which can cause them to swell and in some cases lyse. The result is really an artificial drop in viable cell density. From your perspective, from a customer perspective, that can lead to underestimating cell concentration, misinterpreting culture health, potentially making incorrect process decisions, whether that's around feeding strategies, timing of harvest, or even scaling decisions. And on top of that, the variability introduced by timing differences means that the data may not even be consistent from run to run, making it difficult to trust the measurements or trend performances over time. And beyond osmotic effects, manual dilution workflows also introduce additional common sources of errors, like pipetting variability, dilution inaccuracies, inconsistent mixing, and even incorrect dilution selections. So what may appear to be small procedural differences can actually translate into meaningful impact on data quality and decision-making. This is really what makes our technology different. That onboard automated dilution combined with immediate analysis not only minimizes osmotic shock effects, but also removes many of these common sources of error entirely. And that's exactly what we've done when we built our latest technology, which is the Bioprofile Fast CDV. To date, there are no other technologies out there that truly offer you a fully integrated onboard automated dilution. Even some of the newer systems in the market are have relatively low upper measurement limits, sometimes as low as even 15 million cells per milliliter. But with modern cell lines and media, it's almost common to exceed that threshold early in your process. And in many cases, you reach densities well above 100 million cells per milliliter, which means for most systems, manual dilution isn't an occasional process. It's required for a large portion of the processes you're running. And therefore, with that comes the risk that we've discussed. So as I've mentioned earlier, the recent addition to our portfolio, the Bioprofile Fast CDV, is a standalone cell counter designed to deliver fast, accurate measurements for both cell density and viability across a wide range of cell culture applications. Like our other automated cell counting technologies, Fast CDV requires no manual pre-dilution, and that's important, as I've just spoken about. The onboard dilution step occurs immediately, for as little as 10 seconds after, and that short window is critical. It prevents cells from fully responding to any changes in osmolality, avoiding swelling, potential lysis. In contrast, when samples are manually diluted, there are some delays that can lead to significant impact on accuracy. Now, with the Fast CDV, quite as the name suggests, it's really designed to be fast. The speed's at its core. So you can process more than 45 samples per hour, where you're using the load and go carousel, which holds up to 32 samples, or a 96-well plate format. So really supporting the flexibility that comes with the different types of ways and volumes of tests you may have in your lab. And this and the way the 96-well plates were designed have really ensured consistent mixing and comparable results, irrespective of the format in which you choose to test. Fast CDV also incorporates fully automated onboard dilution, and that's for a wide range. So no manual pre-dilution required, and you can go from as low as 50,000 cells per milliliter all the way up to 140 million cells per milliliter. In addition, the system leverages advanced AI-driven image analysis software to automatically identify and classify cells with a high degree of accuracy. This allows for consistent detection of viable and non-viable cells, even in complex or high-density samples, while reducing user subjectivity and the need for manual adjustments. The combination of automated dilution and the AI-based cell recognition really ensures that results are not only fast, but also highly reproducible and reliable, because we understand your methods are constantly shared across sites and locations globally and labs. So you're able to achieve that reproducible reliability at a higher throughput with minimizing hands-on time and truly eliminating many of the common sources of variability associated with traditional cell counting workflows with as little as 100 microliters of sample. When our development team, we really looked at the product, we closely wanted to ensure and partner with customers and test as many varieties of cell types that we could. This includes well-established cell lines such as CHO, hybridoma, SF9, Vero, HEK, as well as many others. We also extended this work into emerging applications, including alternative proteins, covering cell types derived from beef, pork, chicken, egg, even shellfish. And the goal has really been to ensure that regardless of the application or cell type, the system delivers consistent, accurate results that you can really rely on. The Fast CDV's high capacity and plug-and-play reagent packs are also designed to take the benefit of having throughput and not having to compromise on downtime and maintenance. So each reagent pack supports up to 500 samples, which is leading capacity in this market. And once you install it, you have a use life of up to 90 days. The packs also offer a 18-month shelf life, and making inventory management, ordering very predictable and convenient. Overall, the whole design of the product is to streamline your workflow, reduce operational burden on the lab. And one of the things customers really appreciate about the Bioprofile Fast CDV is that you don't have to choose. You don't have to choose between flexibility and throughput. You get them both. The system offers 2 sampling options, as I said, which fits seamlessly with the different workflows, especially if you're in early cell line development and all the way to manufacturing. From a day-to-day work or even smaller batches, the 32-position load and go tray makes things simple. You can keep adding samples while analyzer is running. There's no need to stop and restart. And when you need higher throughput, you can switch to the 96-well plate format and process larger batches efficiently with a full plate completed in just over 2 hours. What's important is that you're not sacrificing consistency when you switch between the formats. Nova designed its proprietary 96-well plates to match the geometry of the load and go cups, so you get the comparable mixing and reliable results either way. So whether you're running a handful of samples or scaling up to larger studies, Fast CDV really gives you that flexibility to handle both without changing your process or compromising your data. So what does this mean for you and your day-to-day work in the lab? The key takeaway here is that something like Osmotic Shock, which is real, it can have a significant impact on your cell count accuracy, especially if your workflow relies on a manual sample dilution. Anytime you dilute a sample, you're potentially changing the osmolality of that environment. And if that change is significant, cells can respond almost immediately by swelling or lysis. The result is an artificial drop in viable cell density that doesn't reflect the true state of your culture. So if you're using a system that requires manual pre-dilution, it becomes critical to understand that osmolality of both your culture and your dilute, and to minimize the time between dilution and analysis as much as possible, because any even small delays can introduce variability and impact your results. In practical terms, this also limits your ability to batch process samples, because the longer samples sit after dilution, the greater the risk of osmotic effects altering your data. And ultimately, that can affect how you interpret your culture performance and make decisions. This is where integrated solutions can make a real difference with systems like the Bioprofile Flex 2 and the Bioprofile Fast CDV. But that means no manual handling step, no extended incubation times, and significantly less risk of osmotic shock impacting your measurements. So instead of having to manage and control for these variables, you can remove them from the workflow entirely and have a greater confidence in the accuracy and consistency of your cell counts across all the conditions. We've had a very comprehensive comparability report that looks at performance of the Bioprofile Flex 2 CDV module and the Bioprofile Fast CDV against other analyzers like the CDEX high-res or other cell viability analyzers, which are tri-band blue exclusion based. This report goes into detail on how the results compare across a range of conditions, and it's a great resource if you're evaluating different technologies or looking to better understand performance differences. The report is available for free of charge on our website if you're interested, so you can request a copy, and we'd be happy to connect with you and walk you through this. With that, we've come to the end of this webinar. I really appreciate you for joining us, and if you have any other questions, or if you need more information about our products, feel free to visit us at novabiomedical.com. But for those of you still with us, I'm happy to take a few questions. Thank you.
Thank you, Shweta, and thank you for that presentation. Looks like we have a couple questions. The first question is, my cells are pretty robust. How can I be sure that there is a viability drop-off based on osmolality?
Yeah, it's good to know. It's good to have robust cells. It's maybe good to still test to see how osmolality differences play a role for viability and accuracy over time. And the right way to go about this is kind of similar to the way I presented our investigation, would be to have our applications team, our global applications team, sort of work with you and perform some on-site testing and determine how any osmotic shock really, how does that impact and how does that work with your cell line.
Great, thank you, Shweta. The next question, how would I troubleshoot to know if it's the robustness of my cell line or method of testing preparation that is causing these issues?
Yeah, it's a good question. So it's always tricky because it could be one or the other or both in some shape or form having an influence on this. But the best way is to do the tests, is to investigate by trying different conditions and variables, and would be to have a partner with you to help support that. So our applications team have significant experience with benchtop work in the labs doing what you do, and they were a great resource to have on site to partner with you and to do some of this work as you decide which could be the driver for the impact it's having on your workflow. So I'd say it is a trial and error, and we have to partner together to help identify that.
Great, thank you, Shweta. Next question. When you say the Fast CDV has AI capabilities, do you mean there are features that allow for supervised and/or reinforcement learning for live and dead cell detection, or something else?
Yes, that's a great question. So yes, the Fast CDV does have AI embedded software. And what that means is we have used supervised learning as part of our cell detection algorithm that's on the device. So the instrument is able to really look at an image the way our human eye does and be able to identify all of the cells, not just focusing on just the pixel strengths, which is what most people in most devices in the market do. And one of the things that we're really focused on as a business is to empower and push that AI capability further, because there is more that we can do in terms of training and building out the neural networks here based on the different cell types as well. So there'll be more to come with the Fast CDV.
Great, thank you, Shweta. Next question, is Fast CDV a cell counter only, or does it also measure metabolites?
Great question. So Fast CDV is a dedicated cell counter. It does not measure any of the metabolites or chemistries. A fully total combined instrument is our Flex 2. A Fast CDV is really a cell viability density analyzer and counter, and it's if you're to compare it to something else that was in the market, it's similar to your, it's not similar, it's better than your CDEX high-res that you may have in your labs that's being sunsetted and going out of support. And what's unique with the Fast CDV, in addition to just being fast, as the name suggests, is the ability to get that flexibility and the throughput for just your cell image analysis, right? Not worrying about the chemistry, metabolites, etc. And in addition to that, if you are interested about the Fast CDV more, we do have virtual seminars that we host on the Fast CDV and on the Flex 2, where we go a little bit more into depth about both of these products. And if you want to learn more specifically about the Fast CDV, please just let us know. We're happy to have a dedicated demo just for you and your team and go over the images as well as how they compare maybe to what reference analyzer you may have in your lab.
Great. Thank you again, Shweta. I think we have time for one more question. It's, will the CDV packs or the Fast CDV be interchangeable with the Flex 2?
Good question. For those labs that have the two, I'm sure you would like to ordering to be able to swap them, but unfortunately, no, they cannot be interchanged. We have assurance tags, RFID tags to make sure the lot numbers and information are tracked, and so they have separate part numbers.
Thank you. And thank you, Shweta, and everyone for joining us today. If we didn't get to your question today, we'll follow up with you offline. As a reminder, a recording of this webinar will be sent to you within 24 hours of the conclusion of the event. We hope you have a great rest of your day. And once again, thank you for joining us.
Thank you, everybody. Appreciate it.